Take pencil. Hello, good evening to all of you. Today our topic is oxidative phosphorylation. It is one of the important topic. Okay, oxidative phosphorylation. What is oxidative phosphorylation? We will be studying here. What is the concept of oxidative phosphorylation? Before starting about the oxidative phosphorylation here, so we'll discuss about the plus subscription here. So you have convenient plans yearly and monthly. So I advise you to take subscription of Unacademy education. So this is giving a right opportunity to understand. So I avail that opportunity, and this is a very good platform so that all of your doubts will be clarified here. In what way you will be benefited out of this? So here, doubt to clarifications here, doubt clarifications in all topics, and regular quizzes will be conducted, and discussion over complete topic. Okay, so that you will be made perfect in attempting answers in competitive examinations. Okay, about myself here. About myself, I am Anand Babu. I have experience of 15 years. I am qualified with MSc BA. I did my MSc in botany stream. My referral code is Babulai. On my referral code, you will get 10% discount. Now, shall we start our program? Okay. So here, one point you have to remember. All organisms need energy. Okay, energy available in the form of ATP only. Energy is available in the form of ATP. Okay, in all organisms, source of energy is ATP only. All organisms should get energy. Right from the unicellular organisms, the form of energy is ATP only. How this ATP is generated? ATP is very important to carry out all cellular activities or functioning of the organism, without which organisms cannot function here. So this ATP will be generated only by the process of cellular respiration here, by the process of respiration. Cellular respiration is an enzymatic process in which sufficient amount of energy in the form of ATP is available. You see here, 686 kilocalorie of energy is produced. Okay. It is the equation for what is that aerobic respiration here. In aerobic respiration, you see here, glucose getting oxidized here. Glucose is getting oxidized. Okay. End products here are produced, carbon dioxide, and here carbon dioxide and water. And along with it, energy is produced. Some amount of energy is important for the synthesis of ATP molecules. So here is the aerobic respiration, where carbohydrates getting oxidized here, I mean in the form of glucose. Here you see, we are now studying about the aerobic respiration. I mean aerobic respiration, when does aerobic respiration take place? Here first glucose will be converted into pyruvic acid, I mean it will be oxidized into pyruvic acid. If oxygen is available here, okay, oxygen is available, aerobic. Here you see, all of you, there is some problem, electricity problem. Sorry for the inconvenience. Here, ethyl alcohol and carbon dioxide are produced. Here you see, this is anaerobic respiration. This is anaerobic respiration. Okay. So this is anaerobic respiration. But when compared to aerobic respiration, the amount of energy that is released in aerobic respiration is more. So anaerobic respiration is more. Here you see this is. But what is the important point here? Very important point here. In aerobic respiration, oxygen is involved, you know very well. So here energy is produced more. So that is how many ATP molecules here are produced. Sorry. How many molecules here are produced here? See here. In aerobic respiration, 30. Here you see ATP molecules. Here you see. So here ATP energy is formed here. So this is oxidation process. Oxidation process. I mean this oxidation process is nothing but aerobic oxidation. Okay. So how ATP molecules are generated here? This here our topic is oxidative phosphorylation. In oxidative phosphorylation, what is happening? You see, inorganic phosphate combined with ADP forming ATP. So addition of this inorganic phosphate to ADP to form ATP, it is called what is that? Oxidative phosphorylation. Here oxygen is involved. Oxygen is involved here. So in presence of oxygen, this phosphorylation takes place. Ultimately, here these ATP molecules are generated here. Okay. This is oxidative phosphorylation. Whereas in photophosphorylation, here also ATP molecules are generated, but this is related to what is that photosynthesis. In photosynthesis also ATP is generated, but here phosphorylation occurs in presence of light here. So it is called photophosphorylation. This is what is called oxidative phosphorylation. Okay, you see here photophosphorylation, light energy is utilized required for phosphorylation. Respiration, energy of oxidation reduction. So here, what is that? Oxygen is involved. That point you have to understand. Okay. And how this oxidation phosphorylation happens, we will study. But one point you have to remember. Centers for aerobic respirations are center for aerobic respiration. What are the centers here? So mitochondria here. So in mitochondria, oxidative phosphorylation happens in inner membrane. So this is the inner membrane. This is important. Here only oxidative phosphorylation happens. So mitochondria are responsible for oxidation of glucose and synthesize, release the energy in the form of ATP. So this is what is called phosphorylation. Phosphorylation is nothing but addition of inorganic phosphate to ADP to form ATP. So this is very clearly observed here. Next. So in mitochondria, what is the inner membrane here you see? This is the inner membrane. This is mitochondria, and here This is inner membrane here. It's called matrix. This is matrix. Okay. And this is a intermembranal space. Membranal space like this. So here you see, this is the inner membrane. So here inner membrane, one 80 page enzyme is present. 80 page. Here 80 page enzyme is present. Okay, here. Like this, sorry. Here like this. 80 page enzyme is present. So this, what is the important function of the 80 page? What is about the structure of the 80 page? If you observe the structure of the 80 page here you see 80 page. This is the structure of the 80 page here. It has got how many parts here? This is FO. This is FO. And this part is F1 part. F1 part. Both FO and F1 part are connected by protein subunits here. A and B protein subunits. This is A subunit and this is the B protein subunit here. Both are connected like this. And here if you observe the FO part here, 12 protein subunits are present. 12 subunits. All are C like this. 12. Out of 12, 10 are present in the F0 and 2 protein subunits help in the connection of this FO with the F1. Here you see this is the connective link. So these F1 and FO held together by AB subunits. This is A subunit, this is B subunit. Okay. So one point you have to remember. This is fixed in the inner membrane here. This is the inner membrane. Inner membrane. So FO part is in the inner membrane here. Set in the inner membrane. So this F1 part is there now. It is protruded to matrix here. This is the matrix. What is that? This is the matrix of mitochondria. Okay. So this is FO. That point you have to understand. If you observe the structure of this F1 here, the F1 is called a catalytic site. It is catalytic site. Catalytic. So it is very important for the synthesis of ATP. That's why it is called catalytic site. Structurally, this F1 consists of alpha beta protein subunits. How many alpha beta subunits are there? 3 pairs of alpha and beta. 3 pairs. Okay. 3 pairs of alpha and beta subunits by which this F1 of the ATPase is made. This is nothing but what is that? ATPase. Okay, ATPase. ATPase or ATP synthase. Both are same here. So ATPase consisting 2 parts here, F0 and also F1. F0 is made by 12 subunits here, protein subunits, of which A and B subunits connect the FO with F1 here. One more difference, and also here both FO and F1 here have connection with what is that, gamma subunit also. So the gamma subunit making contact, interaction of this FO with the F1 here like this. So this is what is that? Gamma. That point you have to, this is gamma. Okay. And also one more difference between these two F1 and FO. This is able to rotate. FO is able to rotate. This is also called oxisome. It is the smallest particle, rotatory particle in the mitochondria. So it is able to move. It is able to rotate. And gamma protein subunit also moves along with the C protein subunits. FO subunits. FO subunits subsequently move, and this makes movement of this gamma subunit also. But here F1, which is projected into the matrix, it will not rotate. So it is a constant. It is only the catalytic site here. What is the meaning of the catalytic site? This is the place which is very important for the synthesis of what is that? Synthesis of ATP here. So which part of the here oxisome or ATPase or ATP synthase is important for ATP synthase. ATP synthesis means here this is F1. That point you have to understand here. Okay, how these ATP molecules are generated? So this is the ATPase present in the inner membrane of the mitochondria. Okay, how this will synthesize the ATP molecules here? Let us study. Here you see, if you clarify here mechanism. Here is the mechanism of membrane. Here is the linked ATP synthesis explained by chemiosmotic hypothesis. Chemiosmotic hypothesis. This hypothesis is called chemiosmotic. And also here energy released here during the ETS electron transport system synthesizing this ATP with the help of the ATP synthase. Are you following? ATP synthase consists of what? How many? Here this is two components. One is here F0 and another one is F1 here. Okay, are you following this? This is F1. And let us study now how ATP molecules are generated with the help of the ATP synthase. So for studying synthesis of this ATP, one should know about the electron transport system here. This is the electron transport system. This is the electron transport system here. Okay. Electron transport system. You see here in electron transport system, this is inner membrane here. This is inner membrane. Okay. This is inner membrane. Okay, it's not appearing this arrow mark. This is inner membrane. Here you see 4 enzyme complexes are present in the inner membrane. This is what is called intermembranal space, and this side is what is it? Matrix side. This is inner membrane. This is inner membrane. This is intermembranal space here, and this is matrix here. That point you have to understand. So how ATP molecules are synthesized? Before knowing that, here we have to study about what is it? Protons. How protons are pumped from matrix to the intermembranal space. Here this is the matrix now. This is the matrix here. Okay. This is the matrix. Here this is. So this is inner membrane here. One enzyme complex here, NADH dehydrogenase. This is the first enzyme complex. Another enzyme complex is succinate dehydrogenase. Here this is second complex. Another complex here is what is that? Third complex. That is called cytochrome BC1. And another one here is what is that? Cytochrome oxidase. These 4 enzyme complexes come under electron transport only. But this does not come under electron transport. This is ATP synthase. So this is important for synthesis of ATP only. But how electron movement will be there here? You see here, this is NADH molecule here. You know very well in Krebs cycle here, you see, in Krebs cycle, okay, how many NADH, here the NADH molecules are produced, and also FADH2 molecules are produced, FADH2, okay, FADH2. So NADH and FADH2 molecules are generated in Krebs cycle here by oxidation of what is that? Pyruvic acid. And here this is first complex NADH dehydrogenase here. So NADH molecules get oxidized here. Because of the oxidation of NADH here, you see here electrons are moving here accepted by complex 1 here. Through complex 1, the electrons are transferred like this through this direction. Flavin mononucleotides and also iron sulfur, and finally it is received by ubiquinone here. One point you have to remember, this ubiquinone is present between, you see here this is a ubiquinone. This ubiquinone is a mobile carrier here. I mean it moves, it is not constantly present here. It moves from one complex to another complex. But one point you have to understand, this ubiquinone is present in between what are the complexes, enzyme complexes. It moves, here you see complex 1 and complex 2, okay, and also complex 2 to complex 3 here. Here ubiquinone Here also ubiquinone. So ubiquinone moves between complex 1 and complex 2, complex 1 and complex 2, and complex 2 to complex 3. That point you have to understand. This is a mobile carrier. I mean, it picks up the electron from complex 1, okay, like this, and transfers this electron to complex 2 here, like this. This is in this direction electrons move. Already I mentioned here movement of the electrons here, complex 1 to ubiquinone, ubiquinone to complex 3 here. Okay, complex 2 is here, what is that? Succinate dehydrogenase. That point you have to understand. So just like NADH oxidation, FADH2 also oxidized. But oxidation of FADH2 starts with complex 2. That point you have to remember. So here also electrons are removed. Those electrons carried by this FeS, iron sulfur, and also it is received by this ubiquinone. I mean, ubiquinone receives electrons from complex 1 and also what is that? Complex 2. That point you have to understand. And these electrons move here. Okay, finally, I mean, this reaches where this carries the electrons to cytochrome BC1. Cytochrome BC1 is a complex 3 here. Ubiquinone only here. And then what happens here? Okay, and these electrons moving here from here, you see, these electrons are moving from complex 3 to complex 3 to here, complex 3 to complex 4. Complex, this is complex BC1 complex, electrons cytochrome B and FeS electrons, and these electrons like this and find their way, and finally cytochrome C1, and finally cytochrome C. Cytochrome C receives the electrons from complex 3 here. But cytochrome C, just like what ubiquinone here. So it is also what is that? Mobile carrier here. So this is moving between the complex 3 to complex 4. That point you have to remember. Complex 3 to complex 4 here it is moving. This is what is called cytochrome. Just like ubiquinone, the cytochrome C also here receives the electrons from complex 3 and transfers the electrons to complex 4 here. The complex 4 is cytochrome C oxidase. The cytochrome C oxidase here contains 2 copper center. This is one copper center, this is another copper center. And also here proteins cytochrome A and 3 also are there. So the electrons transfer, make their way like this, and finally the electrons move like this. That point you have to remember. Copper center here, electrons move. Okay, finally these electrons reach where? Tell me. These electrons reach the oxygen here. You see here oxygen, and resulting in the formation of what? Water molecule here. So in this way water molecule is produced. Okay, but one point you have to remember, not only electron here, protons also required for the formation of this. Okay, so here important point is how hydrogen here these protons are removed from matrix. Here you see, whenever this NADH oxidizes, what happens? Electrons are removed. Okay, during the flow of these electrons, what happens is some amount of energy is released. Here some amount of energy. Okay, this amount of energy pumps the what is that? Protons which are present in the matrix side here, these protons, into intermembranous space like this. Here you see, during the flow of electrons from, through complex 1 here, 4 protons are pumped from this matrix to intermembranous space here. 4 protons here. Okay, just like that, when NADH molecule oxidizes, these electrons removed now, the electrons transferred to ubiquinone. Ubiquinone transfers these electrons to complex 3 here. That point you have to understand. Here also here, during the movement of the electrons here, some of the protons, like these 4 protons here, are pumped from matrix side to other side. Here matrix, this is a matrix side. This is a matrix side to, here you see how these protons are moving. These protons are moving in this direction. Okay, from matrix to intermembranous space. And also electrons are transferred to cytochrome BC1 to cytochrome here oxidase, that is a complex 4 here. So here cytochrome C receives that electrons from complex 3 and transfers these electrons to complex 4, that is cytochrome C oxidase here. This is also mobile carrier, already I told you here. So it is not constantly present in inner membrane. It okay, it is free. Outer membrane, outer surface of the membrane, it will be present. Here also movement of electrons are there. So here also some protons are removed. So here these protons, how many protons? 4 protons, but 2 protons are important here for the formation of this water molecule. So here only 2 proton molecules will be remained here. 2 proton molecules only remain here. Okay, out of 4, 2 will be utilized in the formation of water molecule along with the electrons here. Protons also required now. That point you have to remember. So for the oxidation of 1 NADH molecules, how many protons are liberated here? I mean, protons are pumped into this. 4 plus here 4. Okay. Here 4. Here 4 is E. And another 4. Here is the 4, 8. 8 plus 2. 10 protons are pumped from matrix to the intermembranous pair. Here concentration difference arises here. I will tell you afterward. Just now listen another important point. As far as this FADH2 concerned here. FADH2 here. Here is the FADH2. So what is about this FADH2 here? Here is the FADH2. Here FADH2 here. So this is here you see FADH2 oxidation starts from this second complex onwards. Here is what do I mean here also electrons move to move through the complex 2 to ubiquinone. Ubiquinone receives electrons from complex 1 and also complex 2 here, and ubiquinone transfers these electrons from FADH2 to here complex 3. Complex 3 to complex 4 like this. So this is a way by which the electrons are moving from one complex to another complex. But here, while during the flow of electrons through this, what is it? Complex 2 here. No protons are pumped here. Sir, why protons are not pumped here? No protons here. The reason is here, during the electron movement here from what is that FADH2, that is complex 2 to this ubiquinone here, I mean this what is that complex 3 here, energy is released. That energy is not sufficient to pump the protons from matrix to intermembrane space. That amount of energy is not sufficient. Okay, amount of energy released at complex 1 is sufficient to pump the protons from matrix to the intermembrane, but here the energy is not sufficient. That is the reason here no protons are pumped from matrix to here. And here during the flow of electrons, movement of electrons through complex 3 and complex 4 here, 4 here protons are formed, and also here 4 protons are formed, but out of 4 protons 2 are used again here in the formation of water molecule. So here only 4 plus 2. That point you have to understand. I will show you. Here you see this is the concept here, and here this is the concept. Okay, this is the concept here. So all points are given here regarding the electron transport. Okay, these are all very important points here. Okay, I will tell you here. So energy calculation here you see. For the oxidation of one molecule of here, one molecule here for the oxidation. One molecule here of oxidation here. Cursor movement. So here you see one molecule of here you see 1 point. For the oxidation of one molecule of NADH, 10 protons. You know very well 10 protons here. Okay, here is the 10 protons. And 1 ATP, for the formation of 1 ATP, how many protons are required here? 4 protons. Am I correct or not? But here, number of ATP molecule generated 10 by 4. 10 means here, for the oxidation of one molecule of NADH, 10 protons. Here 4 means 4 protons are required for the formation of one ATP molecule. So 2.5 ATP molecules are generated for the Oxidation of one molecule of NADH here, 2.5. That point you have to understand. 2.5 here. And next coming to here, one molecule of FADH2. Here how many? 6 protons are liberated. I already told you. Due to the movement of these electrons, protons, how they are pumped from matrix to intermembranous space. Here 6 protons only. So number of ATP molecules here, how many number of ATP molecules here? 6 by 4. Am I correct or not? For the formation of 1 ATP molecules, how many? 4 protons are formed. So divided by 4 is equal to 1.5 here. 1.5 ATP. Okay, 30 ATP, it is equal to 30 ATP. According to old model here, 36 ATP in your textbooks also will be given. But according to recent studies here, here only this is 3 ATP actually given in your textbook. According to old method here, 3 ATP. But actually here 2.5. Here 1.2 ATP are given, but number of ATP molecules only 1.5 according to these calculations. So this is the information here is given. You see, oxidation of one molecule of NADH here. How many molecules of ATP? 3 molecules. This is old method. Oxidation of one molecule of FADH2 and here 2 ATP. Actually here how many? 2.5. Here 1.5. Okay, this is a new method. So recently it has been observed. So this is. Another one is, although aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage, only in the last stage of the aerobic respiration. Am I correct or not? Oxygen is used. Oxygen is very important, without which aerobic respiration will not take place. But oxygen is utilized in the last step of the aerobic respiration. How oxygen combining with the electrons and protons already I shown in the form of flow chart. Okay, some of the protons and electrons react with oxygen and form water molecules like this. So terminal usage of the oxygen here. So terminal aerobic respiration is also. But the presence of oxygen is vital because it drives whole process by removing hydrogen from the system. Hydrogen means protons here. So O2 acts final hydrogen acceptor here. So here you see in this equation, oxygen, 2 protons here, 2 electrons and water molecule is generated. Okay, this is very important process, water molecule generated here. Okay, and next coming to here, you see here this is the diagrammatic representation. Very clearly you can see this how electrons are moving from one complex to another complex here. One complex to another complex here. This is first complex, this is the second complex, and this is third complex, this is fourth complex here. Okay, and this is fourth complex. This is fourth complex here. Okay, this is a fourth complex. Matrix to intermembranal space. Matrix to intermembranal space. So this is matrix. Some protons here pumped into intermembranal. Here concentration difference arises. Intermembranal space concentration is now high. High concentration to low concentration here. What happens? Protons move. How these protons move I will tell you here. And here directly these protons cannot move through this membrane here without any facility or arrangement here. Why? Because it is a membrane, hydrophobic membrane here. Only one is the way. What is the way here by which protons are moving here? I will tell you. So here you see. So this is ATP synthase here. In ATP synthase, what is happening here? What is happening in ATP synthase? Okay, here you see the protons come like this. So this is here what is it? The protons here are formed. Okay, protons. So these protons are pumped like this. So here you see protons, how these protons here, concentration of protons more. Here this side, intermembranal space this is. And these protons are moving like this through connection between these, what is that connection? F0 and F1 here like this. Sorry. Some cursor movement is there. Here you see protons are moving like this. So due to movement of these protons through F0 here, this will rotate. This will rotate like this. This will rotate. Okay. And because of the rotation of this, this gamma part, which is making connection here, you see, between the FO and what is it? FO. Between FO and F1 here, this is cursor movement here. Where it is cursor movement? So F1 here. So here this is F1 and this is F1. So this is a catalytic side. So only this moves. What is this? F0 moves because of the movement of this protein subunits here C, and this makes a rotation of the gamma also, which is a connection between the F0 and F1 here. Okay, then what happens? It will not move. F1 will not move. It is only the catalytic center. Here already I told you 3 pairs of alpha beta protein subunits are there, and what happens here is, what happens here? ATP synthesis takes place. I mean, here you see ATP generated here, phosphorylation happens because of the movement of these protons here. Protons move. These protons, movement of protons will cause conformational changes in the alpha and beta subunits here. Conformational changes will be taken place here. Because of these conformation, structural changes, what happens due to movement of these protons from F1, F0 to F1 here. So immediately what happens because of the movement of these protons due to conformational changes in the catalytic site here. And here what is formed here? Ultimately here you see ATP is generated here. And this phosphorylation happens. And this coupling takes place. ATP will be added to inorganic phosphate. Addition of inorganic phosphate to ATP. That is what is called phosphorylation process. This is phosphorylation process. So number of phosphorylation process this is. But this happens in presence of oxygen, so it is called oxidative phosphorylation. Do you know? Do you understand now how ATP molecules are synthesized? ATP molecules are synthesized only due to movement of the protons through ATPase enzyme from F0 here to F0 to F1 here. These protons cause conformational changes here. This is a catalytic center here. This is a catalytic center. Okay. What is the catalytic center here? This is a catalytic center. Okay. This is catalytic center. This is catalytic center where synthesis of ATP molecules will happen. But one point you have to remember, between F0 and F1, which is rotational here. F0 only rotational. This makes rotation of the gamma protein subunit also. But F1 cannot be rotated. It will not rotate. So just it is a catalytic site where what happens due to movement of these protons through F1 here, conformational changes will happen. Okay, already I told you 3 pairs of alpha protein subunits and beta protein subunits are present. So because of this, what happens? Phosphorylation happens. Synthesis of ATP takes place here. So in this way, for the oxidation of 1 NADH molecule, 2.5 ATP molecules are produced. For the oxidation of FADH2, 1.5 ATP molecules are generated. So ATP generation depends on the protons. Am I correct or not? How protons are generated? The answer is only due to movement of the electrons from one enzyme complex to another enzyme complex. But at the second complex, that is succinic dehydrogenase enzyme here, no protons are liberated. What is the reason? Means here due to movement of electron, energy generated is not sufficient to pump the protons from matrix to intermembranous space. Okay, are you do you understand? Do you understand? So this is related. So energy calculation here. So time is up. Okay, next with another important topic I will come before you. Today some inconvenience we faced because of the power problem.